Welcome to today's paper reading. Today's paper is titled, Large Language Models Surpass Human Experts in Predicting Neuroscience Results. This paper was published on the AR14 preprint by Xiaoliang Luo and colleagues from the Department of Experimental Psychology at University College London. In this study, large language models, LLMs, are shown to outperform human experts in predicting neuroscience results. These models are trained on extensive scientific literature, enabling them to integrate and forecast novel outcomes more effectively than humans. To assess this capability, the authors developed BrainBench, a benchmark specifically designed to evaluate the predictive performance of LLMs in neuroscience. The results indicate that LLMs, such as BrainGPT, which was fine-tuned on neuroscience literature, perform better than human experts. Interestingly, when LLMs are confident in their predictions, they are more likely to be correct, similar to human experts. This suggests a promising future where humans and LLMs collaborate to make scientific discoveries. Importantly, this approach is not limited to neuroscience and can be applied to other knowledge-intensive fields. The challenge of keeping up with the rapidly expanding scientific literature is immense. Potentially groundbreaking findings can easily be lost in the flood of articles. The sheer volume and complexity of relevant findings may already exceed human capabilities. One solution proposed is the collaboration between human scientists and machine learning models, such as large language models, LLMs. These models can assist in various scientific domains, including neuroscience, by predicting experimental outcomes. Neuroscience presents unique challenges due to the large number of relevant articles, the noise and unreliability of individual studies, and the diversity of analysis methods. LLMs, trained on extensive scientific literature, aim to address these challenges by identifying patterns and predicting outcomes, potentially surpassing human experts in certain tasks. Figure 1 contrasts backward-looking and forward-looking evaluations in scientific domains. In backward-looking benchmarks, the emphasis is on recalling factual information, such as historical facts learned in a class. These benchmarks are prevalent in scientific evaluations, focusing on retrieving accepted facts for question answering and reasoning tasks. Conversely, forward-looking benchmarks, involve predicting novel outcomes based on past data, introducing two types of uncertainty, aleatoric, intrinsic randomness, and epistemic, lack of knowledge. An example is predicting the winner of the next table tennis set, considering player performance and potential random factors like wind affecting the ball's flight. Figure 2 introduces BrainBench, a forward-looking benchmark designed to evaluate the ability of test takers to predict neuroscience results. The test cases for BrainBench are derived from recent abstracts published in the Journal of Neuroscience, covering 5 key neuroscience domains: behavioral/cognitive, systems/circuits, neurobiology of disease, cellular/molecular, and developmental plasticity repair. Participants, including human experts and language models, LLMs, are presented with two versions of an abstract, the original and an altered version that significantly changes the study's outcome while maintaining overall coherence. The task for both human experts and LLMs is to identify the correct, original version of the abstract. Human experts provide confidence and expertise ratings, while LLMs are scored based on the perplexity of the text passages, with confidence proportional to the difference in perplexity between the two options. In this study, large language models, LLMs, demonstrated superior performance on the BrainBench benchmark compared to human neuroscience experts. LLMs achieved an average accuracy of 81.4%, significantly outperforming human experts who averaged 63.4%. Even when restricting human responses to those from the top 20% of self-reported expertise, accuracy only rose to 66.2%, still below LLM levels. Smaller LLMs, such as Llama 2-7b and Mistral 7b, performed comparably to larger models, indicating efficient parameter usage. Additionally, LLMs showed the ability to integrate information across the entire abstract, rather than relying solely on local context within the results passages. This broader context integration likely contributes to their superior performance. However, LLMs partially benefit from accurate, domain-specific context, even if not directly relevant to the study, suggesting a nuanced understanding of neuroscience domains. Figure 3 illustrates the performance of human experts and large language models, LLMs, on the BrainBench benchmark. In Panel A, LLMs are shown to outperform human experts, with smaller models performing comparably to larger models. Notably, base versions of models outperform their chat and instruct counterparts, which are optimized for conversational tasks. Panel B shows the distribution of test cases across neuroscience subfields, mirroring the distribution of articles in the Journal of Neuroscience, with behavior/cognitive being overrepresented. The average performance of 15 LLMs and human experts is presented, with LLMs outperforming humans in every subfield. Error bars indicate the standard deviation of accuracy. Panel C highlights that the majority of participants were doctoral students, postdoctoral researchers, and faculty/academic staff, with error bars representing the standard error of the mean. Figure 4 illustrates the calibration of accuracy and confidence for both human experts and large language models, LLMs. The figure demonstrates that when human experts and LLMs are confident in their BrainBench judgments, they are more likely to be correct. Confidence ratings are sorted and placed into equally sized bins, with the mean accuracy for items in each bin plotted. The positive slope of the black regression lines for human experts and all LLMs indicates that confidence is well calibrated, meaning that higher confidence corresponds to higher accuracy. This calibration is beneficial for human-machine teams, as it ensures that the confidence levels accurately reflect the likelihood of correct predictions. Figure 5 illustrates the process of fine-tuning a pre-trained large language model, LLM, on neuroscience knowledge. The model, Mistral 7BV0.1, was fine-tuned using LoRa on neuroscience articles from 2002 to 2022, totaling 1.3 billion tokens. The fine-tuning resulted in a 3% improvement on the BrainBench benchmark, as shown in Part A. Additionally, Part B demonstrates a substantial shift in the perplexity distribution of correct responses, indicating that the LLM has specialized in neuroscience. This fine-tuning process highlights the potential of LLMs to efficiently adapt to specific domains such as neuroscience. In this discussion, we explore the capabilities of large language models, LLMs, in forecasting the outcomes of neuroscience experiments. We found that LLMs significantly outperform human experts on our benchmark, BrainBench, across various neuroscience subfields. This superior performance is attributed to LLM's ability to integrate information throughout the abstracts, particularly focusing on method and study design details. When this information was removed, LLM performance dropped drastically. Our analyses suggest that LLMs learn fundamental patterns underlying neuroscience studies rather than memorizing specific items. This conclusion is supported by techniques to determine text membership within an LLM's training set. We also trained a smaller LLM from scratch, excluding preprints and BrainBench items, and observed superhuman performance, eliminating any potential overlap. Looking ahead, LLMs could be augmented with neuroscience knowledge using LoRa, allowing for continuous updates and interactive experiment design. These advancements suggest a future where LLMs assist scientists in making discoveries, provided they stay updated with the rapidly expanding literature. In this study, ethical compliance was ensured through approval from the Experimental Psychology Ethics Board at University College London and obtaining informed consent from all human participants. No compensation was provided to participants. The dataset, BrainBench, was created through a collaboration between co-authors and GPT-4, utilizing abstracts from the Journal of Neuroscience published in 2023. These abstracts were organized into 5 sections and underwent extensive quality control by both human experts and GPT-4. Test cases were modified to significantly alter results while minimally changing methods and background, ensuring empirical differences without logical incoherence. Thank you for watching.